You're on.
Okay. So today's chapter is, or this week's chapters are regarding the complete nervous system. You have actually 3 different chapters to do. I combined them into 3 different PowerPoints. This PowerPoint deals with chapter 13. The next PowerPoint has to deal with chapter 14 and 15. Okay? So first we're going to actually talk about the neural tissue. And then we're going to actually go talk about the brain and the spinal cord and everything. So neurons. Basically, what is neural tissue? Neural tissue is made up of 2 different types of cells. One is neurons, another one is neuroglial cells or just glial cells. Okay? Neurons, what is the function of neurons? Communication. They generate electrical impulses, which are called action potentials, okay, within the nervous system. And this is how they communicate with each other. Neuroglial cells are multiple types, and throughout the lecture, I will introduce you to the different ones, and we'll talk about their functions. At the moment, their general function Now, there are multiple different types of neuroglial cells, and for right now, their function is just protection and support. As we go through the PowerPoint, you will understand the different functions, okay? Now, right here, this is the basic neuron structure that you were supposed to know. So, what is A? Dendrites. Function?
To
Receive signals from other neurons, okay. What is C? Cell body, and the cell body is where you have, it's a brain of the neuron, okay? This is where you have the organelle, the nucleus, etc. What is this long thing? Axon, okay? You have axon in the beginning, axon terminal at the end. What is the function of axons?
to keep the message like going from neuron.
To convey the message to other neurons, okay? Because this is the receiving end, this is the conducting end. And this is the processing part, okay? Now what is these? What are B? They're the neuroglial cells. As you can see, there are different types and their functions, protection, support. Now, to go more in detail, now, neurons, there are different types of neurons, okay? They're classified based on structure and function. So structurally, you have 4 different types of neurons. Anoxic, bipolar, pseudo, unipolar, and multipolar. Now, there are 4 different types of neurons based on their structure, because structurally they look different. Anoxic, bipolar, pseudo-unipolar, and multipolar. Okay? Functionally, they're functioning also that the neurons are classified according to their functions. So there are 3 different types. You have sensory neurons, motor neurons, and you have interneurons. So functionally, there are 3 different types of neurons: sensory neurons, motor neurons, and interneurons. Okay? And we're going to talk about each one in brief. Okay. So now right here are the 4 different types of neurons that we have. Okay. This one is anoxic. What is anoxic? Here is a cell body. And coming from the cell body, you actually have multiple processes. Here's process 1, 2, 3, 4. But from the cell body, a lot of projections are going out, which are processes. But you cannot tell, looking at this, which process is dendrites, which process is an axon. Okay? So that's why it's called anoxic. You can't differentiate or determine between the dendrites and the axons. This one is bipolar. Why is it bipolar? Because here is a cell body, and from one pole you have an axon, the second pole you have a dendrite. So there are two poles. Okay? This pole right here is a dendrite. This pole right here is an axon. That's why it's called bipolar. Okay? Extending from the cell body, there's an axon and a dendrite. Now right here, it gives you the summary as well. This one, what do you think this one would be called? You have a cell body right here. Okay? It's pseudo-unipolar. Because the cell body is not right in the middle, making it two different poles. It's kind of off-centered. So technically, it looks like there is only one process, this one long process. Pseudo means false. It's still, even that it is on the side, you could still say this is one pole, this is the second pole. But because it's on the side, if you kind of take this out, it looks like there's only one long pole. Does that make sense? This is why this is called pseudo-unipolar. Okay? The main difference is that in a bipolar, the cell body is right in the middle. And you could tell distinctly two different poles. In a pseudo-unipolar, the cell body is towards the side. And therefore, it makes it look like there's only one long process. Now this is a multipolar. This is the one that we actually learned in chapter 4. Okay? This is a multipolar neuron. You have a cell body, then you have dendrites, and then you have basically axon. But the dendrites, there are multiple processes. Here's one dendrite, second, third, fourth, so forth. Okay? That's why it's called multipolar. Because one pole has multiple projections. Make sense? So this is multipolar. So, anoxic, you can differentiate between the segments. Bipolar has two distinct, and as you can see, there's only 1 and 2. And basically over here, you have 1, 2, but it makes it look like just one projection. Over here, you have 1, and then you have 3, 4, 5, 6, 7, 8. Okay? Make sense? Easy enough? Mm-hmm. Now, functionally, we're going to talk about the 3 different types of neurons. Give me one type of neuron, functional, sensory. Give me another one. And internet on. Now, before I start explaining you this, let's say that, as I said, this week I need to, Wednesday there's a gathering for my father's birthday. Let's say instead of us going out, let's say I was going to be at home, okay, and I was going to cook for, let's say, 20, 30 people. And I'm usually running late, okay? So I'm kind of running around trying to decorate my place, clean up my place, cook at the same time. And then my sisters bail on me last minute. Okay, they were supposed to come and help me. Now I'm stuck doing all of the work, which happened in my sister's wedding. Anyway, it was fun, but I was just overwhelmed. Anyway. And to make it worse, on the reception, my dress rips from the bottom because it got stuck in the escalator. I was just like, luckily it was black, you couldn't really see at the bottom, but oh my god, I started crying right before it. Anyway, okay. And she doesn't even know what's going on. She's like, why are you crying? And I'm like, don't worry about it, go enjoy your reception. Okay. What is it? So, let's say I'm kind of running around and I'm in the kitchen and I pick up a pot and I don't realize that it's hot. Okay? I pick it up and as soon as I pick it up, I drop it. Okay? Now, how did I know that that was hot? That it was burning my hands? Sensory neurons, right? So sensory neurons picked it up. So sensory neurons are responsible to pick up sensory information. If I'm touching something hot, cold, hard, smooth, vibration, all of that is sensory information, okay? So does sensory information go from brain to the body or from body to the brain? body to the brain. So sensory information picks up sensory information. Sensory neurons pick up sensory information. Example, temperature changes or temperature, pressure, vibration, all of that. Okay? And sensory information goes from the body to the brain. Now, that information got to my brain, okay? And my brain realized that, okay, if I don't let go of the pot, I'm going to burn my hand. It's dangerous, right? So basically, my brain tells my body to release the pot. That was what made me release. What type of neurons were able to make me release the pot? Motor neurons. Okay? So motor neurons allow movement to occur. Motor neurons allow movement to occur. And motor neurons go from the brain to the body. Now, interneurons. What do you think interneurons are? What are interneurons? Yes?
So ones that are kind of everywhere else and kind of connect to each other and send
They connect. So basically, sensory goes towards the brain, motor comes from the brain back to the body. In between them are interneurons. They're connections. Okay, does that make sense? They are found between the sensory and the motor. They're connections between the two. That's what motor interneurons are. They're neither sensory, they're neither motor. They're classified differently. Okay, because they don't pick up sensory information. They don't do motor stuff. They're in the middle trying to convey it's a bridge between the two branches. Does that make sense? So there could be motor neurons, not motor neurons, interneurons going towards the brain. There will be interneurons coming back from the brain. Does that make sense? Okay. Now, let's talk about the neuroglial cells, because there are multiple types. So what are the believe? Roughly 5 times the number of neurons. Now, neuroglial cells have the capacity to basically reproduce. They have a capacity to reproduce where neurons do not. Which is what this stuff. Functions. Again, right now it's just a basic functions. Some of them are framework. Okay? Some of them maintain the intracellular environment of the neurons, where others act like phagocytes or phagocytes, which are white blood cells. Okay, immune cells. Yes?
What do you mean by framework?
I'll explain that when we actually get into that stuff. Okay. Now, our nervous system is split into 2 main branches. C stands for central nervous system and peripheral nervous system. We'll talk more about the 2 branches in the next PowerPoint, but because there are 2 main branches of our nervous system, central nervous system and peripheral nervous system, there are different types of glial cells. Okay? There are 4 different types of glial cells which we find in the central nervous system, and there's 2 types, but I'm only going to make one in the peripheral nervous system. Astrocyte, microglial cell, ependymal cell, and oligodendrocyte. So there are 4 neuroglial cells, 4 different types of neuroglial cells found within the central nervous system. Astrocyte, microglial cell, ependymal cell, oligodendrocyte. Okay? And from now on we have different slides telling you the function and the structure of each one. Peripheral nervous system, I only want you to know 1 out of the 2 is Schwann cell. Schwann cell. Not Schwann, Schwann. S-C-H-W-A-N, Schwann cell. Okay, now let's first talk about the astrocyte. These are the images of an astrocyte. Okay? There's one astrocyte. As you can see, there's cell body and there's projections. These projections are cytoplasmic extensions. Okay? So where do you find astrocyte? Central nervous system. Structure, cell body, or cell body in the middle, with multiple projection. Okay? Multiple projections. These projections are cytoplasmic extensions. So this is an image of one astrocyte. As here, you could see one astrocyte, second astrocyte, third astrocyte, and you could see actually it's very faint, but these astrocytes are connected to each other. These astrocytes are connected to each other via the projections. Like that. Okay, it's very faint. So, basically, what does this look like? What is it creating when all of them are connected to each other via their projections? No, not blood vessels.
Communication.
No. What else? What does it look like it's forming? No. Stop thinking about size. Just what does it look like? Net. Who said net? Okay. Somebody said net. It looks like it's forming a net, right? What do you think a function of a net is? Capture or protection. Okay. So that's what they're doing. So here's one astrocyte, let's say right here with this projection, second astrocyte with this projection. And they're connected to each other, therefore forming a net. Okay? So their function, they form the blood-brain barrier. They form the blood-brain barrier. Basically, there is a barrier, there's a net between the blood and, well, rest of our body and the brain, or the central nervous system. Does that make sense? So if this part enclosed is the brain, everything else is the blood. So they basically create a net. Anything that wants to come inside the brain has to go through this net. They actually have to go through the astrocyte. So you can see right here, here's a blood vessel. Okay? Right here is an astrocyte with its projections. And here is a neuron. Okay? Some of the projections of the astrocyte are actually connected to the blood vessel. Other projections are connected to neurons, and other projections are connected to other astrocytes. If something wants to enter the neuron, it has to go through the astrocyte and then enter the neuron. So basically, it also monitors what comes in and what goes out. Okay? It doesn't let anything escape because, first of all, you have to go through the net that it made by connecting other astrocytes together. Another thing, anything from the bloodstream has to go through the astrocyte to enter the neuron. Okay? This is called the blood-brain barrier. It's a protection mechanism. Yes?
Is it just blood, or are there other things as well?
I mean, other things would be when it creates a net, okay? But it's directly connected to the blood vessels, okay? So anything can go through the blood vessels and enter the brain. It's monitored that way. Anything that wants to enter just without the bloodstream, then there's a net. Does that make sense? So anything that needs to go into the nervous system has to go through the astrocyte or the net. Okay? Does that make sense? So which is called the blood-brain barrier. Because it creates the blood-brain barrier, or because it's connected to the blood vessel on one end and the neuron on the other, it also monitors what electrolytes are entering the neuron. Okay? It doesn't let too many ions go in or out. It monitors it. Because if you don't have enough certain electrolytes, it will cause damage to the neurons. If you have too much of certain electrolytes, again, it causes damage. So it maintains the internal environment of the neuron. Does that make sense? It also creates the framework. That's because it basically makes the net, it kind of creates a framework of the CNS. Does that make sense? Now, microglial cells. Location, CNS. Function, it basically has like a white blood cell. Protection. It defends the neurons against any microbes, so bacteria, virus, okay? It also protects it against any other thing which can be harmful. Meaning certain, let's say a neuroglial cell is degenerating or it's damaged. It will basically, destroy that neuroglial cell, okay? Maybe a certain neuron is starting to damage. It will basically destroy that degenerating or, neuron. Does that make sense? So microglial cell sounds familiar to what organelle? Lysosomes. Because lysosomes were watchdogs and housekeepers, right?
Yeah.
Watchdogs basically defended against any microbes and the housemates cleaned the area. Does that make sense? So, microglial cell characteristic, it is the smallest microglial cell or neuroglial cell. Out of the 5 that I told you, microglial cell is the smallest one. And you could imagine it has to be small because it has to get through the nooks and crannies, right? Some microbes are so small that they tend to go into a small place. And if that's big, it won't be able to get there. Does that make sense? Yeah? Again, it has a nucleus in the middle and it has multiple projections.
So you said it's the smallest neuroglial cell?
Nucleus cell. Out of astrocyte, oligodendrocyte, Schwann cells, other ones, ependymal cells, it's small.
What did you say it has to be smaller?
Because microbes are extremely small and they could get into places which are very small. Therefore, it has to be able to access them. It's like a kid could get into little cabinets. If you want to, get them out, you have to be small enough to go into that space.
So it just protects neurons from being harmful.
Yes. Harmful, it could be microbes or it could be damaged organelles, damaged other neurons, other neuroglial cells. Now, ependymal cell. Location, central nervous system, but to be more precise, they line ventricles. In the next one, you will understand that the brain contains ventricles, okay? And they basically line the ventricle. Ventricle is a space, which a space is made via, this will be a ventricle. It's hollow in the middle, but it's made by a tissue. So if this is the tissue that makes up the walls of the ventricle, they line that wall, okay? And in the middle, it's hollow. It's hollow because, I'll tell you. Now, the function is, so if this is basically a ventricle, they basically line okay? Basically, they line the ventricle. What they do is produce and secrete CSF, which is a fluid called cerebrospinal fluid. So they basically produce and secrete CSF into the ventricle, into that hollow space. Okay? C stands for cerebral, S stands for spinal, F stands for fluid. Cerebrospinal fluid. Okay? CSF. So ependymal cells, they line the ventricles and they produce and secrete CSF. Okay? And this is what they look like. As you can see, this is the wall of the ventricle. They're lining it and then they produce and secrete that fluid. Now, let's talk about oligodendrocyte.